I would like to begin introducing our speakers of the evening. And what I'd like to emphasize is that science, like any other good endeavor, when it's at its best, it's excellent storytelling. So tonight we're going to have two protagonists, Dr. Sam Webb of SLAC National Accelerator Lab and Dr. Felisa Wolfe-Simon from NASA as well as the US Geological Survey. And like all good stories, today we're going to learn about friendship. We're going to have some surprises, and we're also going to have some intrigue. And with that, I would like to introduce tonight's two speakers.
So Sam and I are really thrilled to be here tonight to talk to you. And that was a great introduction. Thanks.
Thank you. So we just kind of wanted to start out a little bit talking about why there's two of us here. So this is a great collaborative story. Felisa and I have been colleagues and friends for about 10 years or so. I was a postdoc here at SLAC. Felisa was a graduate student. And we kind of, in the scientific world, frequented many of the same conferences. And we were both studying manganese in different systems. But the environmental bioinorganic chemistry conferences were kind of a great way to exchange ideas and thoughts about science.
And this idea of environmental bioinorganic chemistry. So basically, a lot of us, we were part of a community, we are a part of a community, that's interested in the way we interact with metals, we being all life on Earth that we know of. And in particular, let me give you a couple examples. Sam and I studied manganese, but something that might kind of resonate with you, why would you study this environmental bioinorganic chemistry? So like you and I use hemoglobin, or heme, which is an iron protein to move oxygen around our blood. Some things like lobsters use hemocyanin, a copper-based protein. And so it's two different evolutionary solutions to the same problem. And actually, when I was in graduate school, a mere student, and Sam was starting his position here, there was a nutritious discovery for cadmium. And in fact, you don't know probably the enzyme carbonic anhydrase, but this enzyme was discovered that obligately requires cadmium. So you and I and everything else think of cadmium as a toxin, and yet this enzyme requires it. And why do you care? Well, this enzyme is one of the major enzymes used by some cyanobacteria in the ocean to pull carbon out of the atmosphere, and it uses cadmium. And so we're interested in this idea of using metals and how life has evolved. They're toxic, but they're not toxic and required. And so Sam and I had been talking for many years about thinking about metals and how life uses them.
And then, about a year and a half ago, there was a conference up in San Francisco. And at that conference, I gave a talk about how we use the accelerators here at SLAC to do environmental research. And it kind of covered a little bit about arsenic. It covered some other mercury experiments that we have done here. And Felisa was at that talk, and she came running out after the talk and going like, oh, I have the coolest idea. And that was kind of how this all began then.
And so what we want to do tonight is tell you the story of kind of where we're coming from in a very big picture sort of way, and then focus it down. We'll talk about arsenic. And also just keep in mind this idea of toxicity versus nutrition versus something some organism might need. And just the way in which we kind of ask our questions, and maybe questions that you might think of too. And with that, I'll hand it over to Sam to start us on this journey tonight.
Thank you very much, Felisa. So we're going to start talking about life and the diversity of life. So when you think about life, you can kind of imagine all of us in the audience, so we're all alive. Then we have things like our dogs. So things we're very familiar with. That's my puppy. You can imagine cows. Those are things that are alive that we see every day. If you have a flower garden, you can think of the begonias in your flower garden. You can think about the things we eat, corn, trees. We're all used to the California oaks on the hillsides of California here. So those are things we see again on an everyday basis. The lobsters that Felisa mentioned in the beginning, they're also yummy to eat as well. And you can think of the critters that aren't so fun, spiders and cockroaches, mushrooms, all these things that you see in your day-to-day life. These are all the things that you usually imagine when you think of life. But when we put this on a tree of life, so this is now an organization based on DNA and evolution about where everything lands that are related, all of those things, well, you're right there in this little corner, in the animal section here. But all those pictures I just showed, all of the life that we see on a day-to-day basis, all falls in this small little corner of the tree of life: animals, fungi, and plants. So it's a whole realm of life that we don't observe on an everyday basis. And that diversity is really kind of interesting and intriguing. So you also have other multicellular and single-cellular forms of life, like the amoeba, the paramecium, things that you may have remembered from your high school days in biology. But again, there's this whole section now of intriguing things, the bacteria and the archaea, these single-celled organisms that we don't think about on a day-to-day basis necessarily. And you can just even imagine the diversity among the bacteria and these archaea if all the things that we visualize are just in this little branch right here. So now we're going to kind of show you a couple of examples of bacteria. So this is bacillus. It's nice little round rods of bacteria. This is an electron micrograph picture of them. Some of them form spores. That's these guys right here. These are things that I studied when I was studying manganese. There's the other bacteria that you're probably familiar with. This is a picture of Salmonella. This is a picture of E. coli. And you can imagine that even though these bacteria are very diverse on that tree of life, they all kind of look the same too. So the diversity that you're used to in your everyday life, the trees, the dogs, the lobsters, they all look different. These here all kind of look the same in a ways. So in addition to these normal things that you see on a day-to-day basis, or you hear about on a day-to-day basis, there's a whole other types of bacteria that you don't hear about. And so there's cyanobacteria. These are bacteria that utilize light for energy sources. You have, this is a Deinococcus radiodurans. This is a radioactive resistant bacteria. So now we're kind of thinking about all the other crazy things that bacteria can do. This is the, let me go back. This is the super bacteria that everyone talks about occasionally that's going to withstand all kinds of radiation damage. In addition, you can think about other environments. So this is now the more extreme environments. So when you talk about the other branch of the single-celled organisms, like the eukaryotes or the archaeans, they tend to be called extremophiles. So they like extreme environments. So this is Yellowstone National Park, Grand Prismatic Spring. And you can see all the colors here, and it's all due to the different biology at different temperatures in the spring. And here you can see a drainage area, again, with different types of bacteria at different temperatures. Not only do you find these kinds of extremophiles in Yellowstone, you can go down to the mid-ocean ridge vents, and you see all kinds of thriving bacterial communities in these really crazy, high-pressure, high-temperature extreme environments. And they're all still single-celled, simple organisms. So again, another crazy diversity of life in a very simple organism again. So these are extremophiles that love the heat. You also have extremophiles that love the cold. So this is pictures of Antarctica in the dry valleys. And there actually is, again, a thriving microbial community that lives underneath the ice there. And they look kind of like this. This is a picture of some microbes isolated from underneath the ice in these crazy environments. To kind of continue the extreme environments, you also have halophiles. So if you think about halophiles, that's people that are bacteria that love salt. This is a picture of the Atacama Desert. And you can see, again, yet another thriving community that really just loves to be in the salt crusts of the desert there. And again, simple, single-celled organisms. So all of this diversity of life that we have in the bacteria and the archaea are all look kind of the same, but it's an incredible diversity. And yet, there we go. And now you can kind of see where all these guys lie here. And yet, all of these forms of life are all made up of the same building blocks of life, the same elements in life. So here's a picture of the periodic table. It's kind of a kind of way of organizing all the elements that we know about. Hydrogen, helium, all the way through here. And elements that have similar chemical properties are organized in columns here. So boron is similar to aluminum, gallium, and so on. So we'll come back to that a little bit later on. But again, all of the life all the diversity of life have all been presumed to use the same major elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And these 6 elements make up all those building blocks of life. So you can kind of imagine building blocks being like, these elements being the building blocks like blocks of Legos. And you can arrange these blocks of Legos in different combinations and different patterns that can make up different biological structures. And so to further talk about some of these more biological structures, I'm going to let Felisa go into more detail about some of the biochemistry that goes on with these guys.
Thanks, Sam. So Sam kind of gave you that overview of all the diversity of life. And what I want to convince you of now is actually all that life you heard of, everything that seems so exciting, there's a unity to it, and there's something that all of it has. And so Sam's introduced you to these 6 elements. So now we'll show you what those 6 elements do. And so to continue the story, it's those 6 elements, C, H, N, O, P, S, in these 3 pieces of the cell. DNA, or nucleic acids, proteins, and lipids. And you've probably all heard about these 3 pieces of every cell that in some form or another. And now this is all cellular life, so we could argue about prions and virus, but for now, let's talk about all cellular life we know of. And here you see on the left-hand side, we're looking at just those different pieces stained. So let's talk about these 3 pieces so you can kind of get an idea of when we say building blocks, that diversity of life, what does it really mean? And so first, the DNA. So I think we're all very familiar with DNA. It's the information technology of the cell, and it's where evolution happens. So this is like the computer code that runs all the cells in your body and in everything else on Earth. And here you can see DNA of that cell that you saw earlier stained in blue. And just to give you a picture of the crystal structure, which was solved on an X-ray beam, here you see the very famous double helix of DNA. So the DNA, that's the information technology. That's where evolution happens. Number 2, proteins. And now I think more familiar to many of us in thinking about protein is maybe the steak you had for dinner. Let me introduce you a little bit to a different, let's get a little more specific about the protein that we know. And so the proteins in a cell, they're the molecular machines and the biological girders. So the DNA codes the information to make the machines. And the proteins in every cell in your body and every cell in a microbe generally either do work, they do some kind of work for the cell, or they hold up the cell. They have some kind of structural component. And here you see again, stained in this particular cell in red, are the proteins. And I want to introduce you to what I consider a pinup, because it's a beautiful protein, I think. This is photosystem 1, and why do you care? And all the plants outside, this is actually from a cyanobacteria, and all the photosynthetic organisms on Earth. And you can take a look up here. We brought some photosynthetic organisms from a lake we'll talk about in a minute. What you're looking at is an incredible feat of evolution. There's more than 36 proteins, and if you kind of squint, you can see the different colors. It's trilobed, so it's a repeat in 3. So there's more than 36 different proteins that all work together to do photosynthesis. And also, you see all these things in yellow. So those are all the cofactors. Actually, it happens to be chlorophyll. So this is an incredibly complex machine, an evolutionary feat of life to be able to make photosynthesis. So those are proteins. They do the work of the cell. And 3, lipids. So this separates you from everything else. And here you see it's stained in green. And so I think we're all very familiar with lipids or fats. Here's just one, I think, illustrative example. This is just Kool-Aid in water with some oil. And you can see that it naturally forms these abiotic vesicles, which kind of take that Kool-Aid and lock it in a little place. So this is an easy experiment. I suggest all of you, say under the age of 15, do it home tonight. So we have those 3 pieces and those 6 elements in those 3 pieces, and we'll keep mentioning that over and over again, in all this life. So all the life that Sam introduced you to, in the coldest of the cold of Antarctica and the deepest of the hottest part of the deepest ocean, all of that life, including you, is made of those 3 pieces. So here we have that put on the tree of life, is made of these 3 pieces. So now what's interesting is, so we just convinced you there was the diversity of life, and then I kind of turned it on its head and said, no, it's actually all the same. But what's the same about this? So let's be biogeochemists for a moment. And what I mean by that is we're going to break it down and get back to those different elements, because you want to know what the elements are in what piece, maybe. So the DNA in a cell is basically the phosphorus. On average, you can think all the phosphorus in the cell is basically being in the DNA, and we'll come back to that. The proteins here you see now appearing in red it's a lot of nitrogen. And so when you see a protein-rich food, it's actually a nitrogen-packed powerhouse. Now the lipids are carbon. So you can think of like the gasoline in your car. That's a carbon-based compound. And so if we want to be biogeochemists for a minute, and we look at, if we want to kind of take a big step back and think of, okay, there's those 6 elements and these 3 pieces, can we really be abstract and squint and see the pattern in nature? We'll kind of see the phosphorus is in the DNA, the proteins are made of essentially nitrogen, and the lipids are made of carbon. So if you want to think of an alternative to biochemistry that we know really well, so everything we just told you, Sam and I, we think we know really well. All those organisms, and we keep discovering new bacteria, and we keep discovering new birds, for heaven's sakes, right? All of that stuff is made of those 6 elements and those 3 pieces. So that's what we know. So we're going to start from where we know and kind of see if we could make it a little different. And so we'll continue to tell you this story about how we might start asking that question of, could you have an alternative to what we just told you? So if we were biogeochemists a minute ago and I told you, or you believed me maybe, that these 3 pieces are made up mostly of those 3 elements, let's start by asking a very simple question. Could we, instead of using phosphorus, say in a major place that we might find phosphorus in the DNA, could we find arsenic? So this isn't just a hypothesis that Sam and I dreamed up randomly. Let me show you where we're coming from. Again, to follow the idea of the logic of, wow, we know all this stuff, but how could you find something you've never seen before? So if you go back to the periodic table that Sam showed you a minute ago, and here highlighted in green are the 6 elements that Sam introduced you to that all life is made of, if you notice that arsenic is just below phosphorus. And as he mentioned also, there's a periodicity, so there's a relationship that the different elements have next to each other. And what was curious to us about phosphorus and arsenic is that, well, I'll only tell you a couple of things, but there's a lot of ways they're really similar. So one thing, for example, is the atomic radius of phosphorus and the atomic radius of arsenic, the atoms, are very, very similar. And they're very different. You see that phosphorus and arsenic, one's right on top of each other. These 2 are literally very similar in size, and yet they're very different than nitrogen, which is one above. Also, they have very similar other types of chemistries, not just their size. So we started to think, well, why is arsenic, how does this relate? And so Sam showed you earlier this idea of the building block, and I think he wants to give you a little bit more taste of the demonstration. If we go back to those building blocks, the analogy that Sam made earlier, in Legos, like Legos, here we have, for example,
there we go.
We're very interactive. The idea of these building blocks, and you'll see there's 6 colors, it's as if we changed out one of the colors that he will demonstrate for you now. And you see that it's not the same blue, it's a little lighter in blue. And the idea is that it's like similar, but just a little bit different. So could it work? It's a very simple question. I don't know. And so this idea of the building blocks that Sam was so fantastically demonstrating, and really thinking about the abstract way life is composed. But how do you know arsenic? When we think of arsenic and old lace, I like to say arsenic and old life, but maybe that's a bad joke. So how we normally think of arsenic is as a toxin. And one of the curiosities, I'm a biochemist, one of the curiosities to me, and Sam and I would talk about, is why is arsenic toxic? So arsenic is toxic because it looks a lot like phosphorus. And that's really fascinating. So it's toxic to you and me and to all sorts of life. It's toxic to everything, even the biology that uses it we'll mention in a moment, because it looks like phosphorus. There's a confusion the cells have. Once it's inside, they go, oh, this is wrong, and they try to get it out. But it looks like phosphorus. And here you can see, in fact, there's a famous story about Paris green, which was a green paint. Now, potentially one way Napoleon may have died. It turns out the craziness of the story is that when you put this paint in certain wallpaper in the 1800s in France, if I have my facts right, and I might be a little confused, a fungus would eat the arsenic and metabolize it in a certain way. If anyone's interested, we can talk about it later, but release an arsenic gas that's really poisonous. And so there was some suggestion this may have been one thing that killed Napoleon. So I think we're very familiar with the idea of arsenic as something bad. And actually, you can see here on the left-hand, or I guess right-hand side, in places like Southeast Asia, the arsenic concentration, the natural arsenic concentration in the water is so high that it leads to lesions on the hands and feet. And actually, it is a huge problem in areas of Bangladesh and other parts of Southeast Asia. So this is normally what we think of arsenic. but it's generally toxic because it looks like phosphorus. So that we thought, well, this is very interesting. So we asked a very simple question. Because remember, we started by saying, life has come up with unique solutions to the same problem, and generally this problem is being alive. That's the problem. You got to solve that, how to be alive. And we told you, iron and copper can do the same thing, move oxygen around. And then I mentioned this discovery that was made a number of years ago now about cadmium, a toxin actually being required for the function of something in biology. So we asked the same sort of question, very simple question. Can a microbe use arsenic in place of phosphorus to sustain its growth? And so what we want to do now is walk you through the story of how we discovered the microbe and what kinds of tests we did on the microbe to be able to do this, to answer this question. And first, what I want to do is tell you kind of how you go about asking. So that was a simple question. We want to test that hypothesis. And where might you want to look for some sort of alternative biochemistry? And just like Sam mentioned, if you want to find a very hot microbe, a biology that can live very hot, you might want to go to a deep sea vent that's well over boiling. If you want to find a microbe that lives very cold, you might want to go to the dry valleys of Antarctica. And so we want to hedge our bets as microbiologists. We want to go somewhere where it's the natural experiment has been set up. So we might want to look for an environment of high arsenic and enrich, and I'll show you what enrichment means in a minute, and kind of hedge our bets to something that could cope with or survive with high arsenic. And then you would want to identify that substitution. Can a microbe sustain its growth using arsenic in place of phosphorus to sustain its growth? So we want to be able to show if it could do that. And so what I want to introduce you to is our field site. And it's a fantastic area. I'll show you some pictures in a moment. And what you'll see, so we're here out in the west. I'm from the East Coast, so I'm still getting used to the sun isn't rising over the ocean. I think the Pacific is amazing, but it's been an education. So what you'll look, if you go east here, just on the other side of Yosemite National Park is Mono Lake. And Mono Lake is a remarkable environment, and it's a critical thing to remember, it's high in arsenic, natural arsenic. And we had the fantastic good fortune to be able to collaborate. And here you can just see an example of his paper, and all you really need to know, it says the ecology of arsenic. Dr. Ron Ormland has spent most of his career, most of my lifetime, studying arsenic. And there's a lot of microbes that actually can use arsenic. But they use arsenic like you and I use oxygen. We breathe in oxygen, breathe out CO2. So these microbes, and it'll be a little to wrap your head around, but they don't live with any oxygen. And they take in arsenic in one form and breathe it out in another. So that idea that microbes and other organisms could live around arsenic was very well studied. So we wanted to go to an environment and collaborate with other scientists who really know certain things really well, how other microbes deal with the arsenic, and also to this environment, Mono Lake, that is very, very well studied. Because we wanted to go someplace where we know a lot about the life there, and ask a unique question. And so when you go out to Mono Lake, it's a really fantastic environment. It's about 3 times the salt of seawater. These are some images that a photojournalist took with us last September when we were out in the field. You see these fantastic tufa formations, which are calcium carbonate. Again, it's very high in arsenic. It's very basic. If you know chemistry, it has a pH close to 10. It's a remarkable environment. And one of the things that's most stunning about it, it's about 100 square miles in area today, but it was over 700 or so in the Pleistocene. So if you haven't been there, I strongly suggest, as being in California, you should see it. So it's very salty, very basic, very alkaline, and it has high arsenic. And this last image I just want to point out is one of my favorite. You can see the moon here bright in the late summer sky, and there's Jupiter shining over Mono Lake. So it's a really remarkable environment. And in fact, you can see, here you can see the Sierras a bit in the back. So if we went out to Mono Lake and we wanted to find this organism, so I told you there were some organisms, that's arsenic, AS is the chemical symbol for arsenic. And there were some organisms that could take arsenic in one form, like we breathe in oxygen, right, and blow off or get rid of the arsenic in another form. And here you see that we're showing it as changing color. But we wanted to find something seemingly a little different. We wanted to find an organism that could take arsenic in one form and take it inside of the cell and use it. And so one of the ways we did this, so I want to show you a little bit of a demonstration, is so here we have a test tube rack And we took some mud, literally, just like the muds you're seeing from Mono Lake right here. And we put a little of that mud, these are just examples, no arsenic, promise. We took a little bit of the mud and put it in the first test tube. And we made what's called a slurry, so this would just be muddy. We let that cook, we say in microbiology. We just let that sit for a little bit of time. And what's interesting about the microbe I'll introduce you to in a minute, it's very boring in all other ways. We just heard about those extremophiles. It's eating sugar, room temperature with plenty of oxygen. So in a lot of other ways, it would be seemingly uninteresting to you. So this is muddy. We've just inoculated that with some mud from the lake. We wait for a while. We take 1/10 of this tube and put it to another tube. And so we've diluted it 1 in 10, and we just let that cook for a while, and just let it sit. In fact, what I've learned doing microbiology in this way is you have a very intuitive, patient set of Zen-like quality. I have a lot of high energy. It's been quite an experience. So you do that, and we do it again, and you do it again, and you do it again. And I've filled the test tube racks like this. And in each of these, you're probably wondering, well, what kind of water did you put it in? And so that's Mono Lake. And we know all the chemical constituents of Mono Lake. And we make an artificial Mono Lake water, kind of like you can buy the seawater, the instant seawater at the fish store, at the pet store. So it's like an artificial, actually this is artificial Mono Lake water, it's actually really. And the idea is that we can control everything. And each one of these is very high arsenic, if this were the actual experiment. And so we're losing the original memory over time in that dilution series of Mono Lake. So any phosphorus, all the other nutrients, I can completely control those nutrients. So by the time we get to this one, so this is some time later, we can take a drop of this and put it on a Petri dish. And these are all safe. You're welcome to come look at it after. And then this Petri dish we let sit. There's a lot of Zen about this. And then little bumps start to grow. And we're going to pick one of those bumps and characterize it, because it has only arsenic for a while. We haven't given it any phosphorus for months. This process takes months. And this is what we found. This is strain GFAJ1. And just like the conditions I told you it grew under, sugar, glucose, room temperature, about 20-some degrees Celsius, 28 degrees Celsius, and aerobic, so with oxygen, nothing like the extremophiles that Sam just introduced you to. But this is what we found. You can see from the scale bar, it's about 1 by 2 microns. It looks almost like a little gnocchi, little potatoes. They don't look particularly interesting. I'll tell you under the microscope, they're real fast swimmers. We're not sure how they swim yet. But this is the microbe we found. And it was more of a, gee, that's not right, rather than a eureka moment when we were in the laboratory, because we just couldn't figure out how it was doing what it was doing. So we wanted to, so we've done the enrichment part. That's the enrichment. And we looked for tolerance, something that could stay alive at the last tube that should have been completely void of anything alive. So we wanted to find out some information about this microbe. So one of the first classical things you might want to do is do a growth curve. So what we want to do is just see how it grows. So you can see time here on the x-axis, and growth measured in number of cells. And it's actually what you're thinking. You take a little bit of that tube, put it on a microscope slide, and count. More Zen-like quality. And so what you see over time, if you add neither arsenic nor phosphorus, you get very little growth. You get a little bit, but it's not, they're just seeming to stay at the same number of cells. Now, dramatically enough, if we add it to a tube with phosphorus, what you see is a dramatic growth, right? A dramatic rise in the number of cells over time. That wasn't entirely surprising. Mono Lake also has quite a bit of phosphorus along with its very high arsenic. Now, what was interesting to us and rather surprising is that when we add arsenic, we also got stimulation and growth. And that's not supposed to happen because arsenic is toxic. Even to the microbes I just introduced you to a minute ago that can use arsenic, breathing it in and breathing it out, it's still something toxic to them. In fact, you have to kind of spike it into the tubes for them to be able to use it because it's still toxic even though they're doing a microbial process with it. So this microbe seemed to be a bit counterintuitive, but it's starting to get to our question. Can a microbe grow substituting arsenic for phosphorus to sustain that growth? So this was pretty compelling that we should at least find out what's going on. So the next thing we wanted to find out is not just that arsenic stimulated the growth, then it was a little bit less than phosphorus, But what was the arsenic doing? And so what you're looking at here is a technique. It's a type of mass spectrometry. But what's important to take away from this image is on the left-hand side, you see there's two kind of fuzzy images, and that's the, just like I showed you here, this is a scanning electron micrograph showing you the different cells. This is like a scanning electron micrograph, so we can see the two cells there. And on the other side, we have the arsenic to carbon ratio. And so all this is really telling you is based on a heat map. So the warmer colors, the yellows and reds, are a higher amount of arsenic in the cells. And the way this works is you have the cell, so if this is my cell, you can think of it sort of like a laser beam. That's almost the right analogy. And it's burning through the cell. So you go down into the cell, and you're going to be able to measure where the arsenic is. So it's not just arsenic stuck to the outside. It's actually arsenic that's been taken up and inside the cells. So arsenic seems to stimulate growth. That was the first thing I showed you. Arsenic now seems to be inside the cells, incorporated into the cells somehow. But now you want to know, well, remember we had those three pieces of the cell, DNA, RNA, proteins, and lipids. Where is the arsenic? Is it just randomly in the cells? Is it associated with these pieces? And so what we found was, we found that it was associated with actually all the pieces. So the way this experiment works, you can think about these test tubes. We can get arsenic in a certain form, arsenate in particular, as a radioactive tracer. So we can radio label the arsenic and then just let the cells grow with it to see where it goes. And because it's radioactive, we can actually follow where it is. So there's a certain amount of the atoms are radioactive. So when the cells take it up and those other things I just showed you, the growth and also the images where it's inside the cell, we can then separate the different pieces of the cell and determine, because which is radioactive has the radio-labeled arsenic. So we can determine that, and that's what you're seeing here. So what we found is there was some of the radioactive label of arsenic in the DNA, some in the lipids, and a large portion in the proteins. And it was at about this point that I saw Sam's talk at a meeting in San Francisco, and I thought, oh, this would be perfect. What he showed in his talk, if we could make those measurements on GFAJ1, it might really be able to help us understand what's actually going on here. And so Sam, I think, will pick up the story now to tell you how we really started to understand the story behind GFAJ1, being able to come here to SLAC to make those measurements.
Thank you, Felisa. So we've told you a little bit about how Felisa found GFAJ1. So now probably the main question in your head is, you have this bacteria that seems to like arsenic, but what does this big accelerator here at SLAC have anything to do with bacteria, right? So here's the SLAC campus. You guys are all in this little corner right here. And there's the long accelerator that everyone's used to hearing about. When you think about particle physics at SLAC, that's what you always think about. But we're going to talk about primarily where I work at SLAC, which is SSRL. It's the Stanford Synchrotron Radiation Laboratory, light source. And this is a different accelerator, so it's different than the really long linear accelerator, but we use it to do all kinds of research, including looking at environmental and biological problems. And it's a circular accelerator just in the corner here. So, you know, I said it was the Stanford synchrotron, and the first thing you go is like, well, okay, that's great, but what really is a synchrotron, right? To make it as simple as possible, a synchrotron is an accelerator that's in a circle, and the synchrotron spins the electrons around in bunches in a circular ring. And its sole purpose then is to create X-ray photons. Because if we spin that electron around in a circle, every time we bend the path of that electron, it's going to emit radiation. If we spin it fast enough, the radiation that's emitted will be X-rays. And so the whole purpose of our synchrotron is X-ray photons. And so here's a little schematic of a synchrotron, and there's the green dots, that electron. It's created in a small linear accelerator. It goes around into a little booster to get it up to the right speed, and it goes around really, really fast. It's not quite the speed of light in this case here, but in the accelerator it really does go very, very close to the speed of light. And every time it goes through a magnet, which is going to bend its path, you're going to watch and see a little photon emit from the little magnets here. So there. And those X-ray photons are going to go into our experimental station, and there we can do all kinds of different experiments, including experiments on our GFAJ1 microbe. This is what it looks like, our little experiment. It's a goofy picture of me. But the X-ray beam is coming in from the right-hand side here, and you see this mysterious plastic box. Inside that box are two mirrors. And those mirrors focus the X-rays down in the horizontal and the vertical direction from the big beam that comes out of the accelerator to a very small 2 by 2 micron beam. So you've heard about 2 microns. That's about 1/100th the size of the width of your hair if you still have hair. Like mine's going away. But in any case, though, we can focus that beam down to a very, very small spot, probe small, small samples, and then look at the X-rays that interrogate the sample and determine the chemical properties. So you're going, okay, great, we have accelerators, we have X-rays, that sounds really cool. But what can I really do with X-rays, right? So we're saying we're going to use X-rays to investigate these bacteria, but what can it do for me? The kind of X-rays that you're probably used to experiencing or hearing about are medical X-rays. This is actually the very first X-ray picture that was ever taken by William Roentgen. My pronunciation is probably a little bit incorrect, but this is his wife's hand. He stuck her in there and X-rayed her. And you can see that X-rays absorb matter, right? And so if they're absorbed by certain areas, they're darker, and if they don't absorb, it's lighter, right? So you can see the outline, and from the medical field, this is very important, right? But what else can I do with X-rays? I'm going to talk about two different techniques that we use. The first is X-ray fluorescence. So this is a little schematic, not quite to scale, of an atom. You can see the center of the atom, all the neutrons and protons, and you have clouds of electrons that circle that nucleus. We take an X-ray photon, it's going to come in there, and it's going to, if it has the right amount of energy, you can think of energy of a photon as being the kind of the color. So if it's strong, if it has just the right energy, it's going to be able to knock out an electron from that very center shell of the atom. Now, that atom now is kind of unstable. It's going like, hey, I'm kind of missing something here. And it needs to relax. It's kind of, after being excited, it needs to relax. Very key terms. And so one of those outer shell electrons is going to come in and fill up that lower vacancy. When that happens, you'll see there's an X-ray photon that's emitted. So this whole process of absorption and the emission on the fluorescence of another photon is characteristic of the element that's being probed. So this way, we can uniquely identify the element that the X-rays are being absorbed by. So this can tell us what elements are where in the sample. And then with that 2 micron beam, we can tell exactly which elements are located where on the sample. So that's pretty fine and pretty exciting. But we can carry this yet another step further. We can do X-ray spectroscopy. So now we have our absorbing atom kind of here in the center. And more than likely, there's probably some other atoms around in the neighborhood. When that photon comes in, it's going to attach here, and now it's going to knock out that electron. Now, physicists are always kind of, have this duality of particles and waves. So now we're going to picture this as a wave. And that wave now is going to be emitted. And you can kind of imagine if you were in a nice still pond and you have a big boulder and you throw it into the middle of the pond. You're going to see little ripples kind of come out from the center. And that's kind of how this electron that's being emitted is going to ripple out from the center. Now, if you happen to have other atoms around, you can imagine them as other boulders. And as that wave propagates out, it's going to kind of bounce and scatter off those other boulders. So it kind of scatters out a little bit. And now it's going to scatter off those other atoms. You're going to see other little bumps form. And those other ripples now are going to continue to propagate. And now you're going to see there's all these really interesting little patterns here. So there's all this interference of all those different ripples. By looking at this ripple pattern, we can then determine what these other neighboring atoms are. So by probing the absorbing atom, we can tell you what's around that and actually tell you about the chemical structure of that absorbing atom, say in this case, arsenic. So what does that look like? So there's a lot of other stuff that goes into this, but this is kind of the final result. You can kind of get an idea of how far things are away. This is the distance in angstroms, very small, and how strong that element scatters. So this would be arsenic and, say, oxygen neighbors. You can see there's a little peak where the oxygen atoms are located. If we compare that to, say, sulfur, we see that the peak is at a larger distance. The sulfur atoms are bigger than oxygen atoms. And sulfur is also heavier. And so the same amount of sulfur will be at a further distance and have a stronger magnitude. So we can tell different things apart from each other. And if you have structure beyond that first coordination environment, first shell, you can also see further things. So this would be if you had oxygen and iron, and we can compare that to, say, oxygen and carbon. And again, you can see there's dramatic differences even in the second shell environment between those two elements, between iron and carbon. Carbon's much lighter, much smaller, and so those differences are quite dramatic. So we can kind of use this as a fingerprinting and fitting tool to kind of look at the environment around the arsenic and tell us what it's bound to. So the next thing here is a movie. This is kind of showing different biomolecules, and we're going to kind of focus in on the DNA since it's a prime example of where phosphorus is in a biomolecule. And there's kind of a roller coaster ride. You can kind of see the phosphorus atoms in the backbone of the double helix here, all the little yellow guys. And hopefully you're not too seasick, but you can come back and kind of get the overall picture now of where phosphorus is in biomolecules like DNA. And the key thing here to recall here then is, there we go. There's our phosphorus. You can see it bound to 4 different oxygens. And you can see if you go a little bit further away, there's some other carbons just a little bit further away. So we're in an environment where we have oxygen and then carbon just bound to it just a little further away. So we probed it with the X-ray beam, and we get a fingerprint that looks like this. So these are the cells of the GFAJ1. And so if we were to compare to it, like what would arsenic happen in most other types of cells? What would happen to arsenic in an environment like the well-characterized arsenic respirators, so the ones that breathe arsenic? Well, they usually take the arsenic and bind it to sulfur to get rid of it. Oh, that's that one. So again, that looks very different from what you see in our microbe. It's a completely different distance, and not at all like what we see. You might say, well, what if it just kind of fell out on other things? That would be if it just kind of co-precipitated or co-fell out with iron. It would look kind of, the second shell, these other shells would look more like this. Again, that doesn't really match. If we take arsenic kind of in that same kind of biomolecular structure, it would look just like this in the red. And so you can see that we have a pretty good match. So we can kind of then compare that. So this fit here is just taking that phosphorus and making it an arsenic. So the data was showing us not only do we have arsenic that's going into a cell, sustaining the growth, but it's also being chemically bound in a way that's very peculiar. I mean, that's completely different than we've ever seen. And it appears to be incorporated into these biomolecules. So we've been kind of showing DNA as the example here, but there's also, again, the proteins and the lipids. And all of the phosphorus in these kind of structures are very similar. Arsenate in these oxygen-carbon esters. And again, also the data that we see kind of shows that it's kind of consistent with all of these phosphorus locations. That's pretty cool. So you might say, well, so you found a bug that grows with arsenic, so what? What's the really cool part about that? And for the kind of the philosophical ending, I'll leave it back to Felisa to kind of wrap it up for us.
Thanks, Sam. I must admit, he did the heavy lifting. That was some pretty serious data and science you just had a taste of. And yeah, so what? And so I think for most scientists, they would tell you, what are we taught? This is what we did, this is what we did, and this is what we did. Tell you again, tell you one more time. But instead of telling you what we just told you, what I really want to get across today, or will be 2 more slides, we're almost there, and the idea that really what we've shown you, it sustains its growth, it's inside the cell, and it seems to be in these chemically consistent environments to where we would find phosphorus, for example, in a structure like DNA. Well, gosh, what does that mean? Really, it opens up all sorts of new questions that not just Sam and I, but all sorts of other scientists need to bear down their gifts and talents on trying to understand what happened. What is this microbe doing? You know, this has generated quite a bit of chatter in the scientific community. And there's good and very interesting criticisms that we're trying to understand. We don't understand. You know, it's not as common to be able to deal with really cool, new, exciting questions in science. And as a scientist, we might call these first principle questions. So we can ask a very simple question. We thought we might get a very simple answer. And actually what we just showed you was, instead of a eureka in the laboratory, it was more like 2 a.m. looking at the tube going, well, that shouldn't be that way. And trying to understand why there was still something growing in that final tube. And so let's leave you then thinking about, we knew so much about life. Where did we start? The diversity of life. And then we kind of did a 180 and said, all life is actually all the same. And actually, could it do something different? We've shown you some interesting evidence. Maybe something's doing something different, some microbe. If you've noticed, so I'm a fellow at NASA, a research fellow, and I'm part of a group of people interested in something called astrobiology. And what is this astrobiology? It's the study of life in a planetary context. And so astrobiology is the study of the origin, the evolution, the fate, and the future of life in our universe. And it's really just thinking about what's going on and what is this life in this planet that we know of. And so what I'd like to leave you with in thinking about astrobiology and how we bared our tools on trying to ask the question here on Earth, I'd like to leave you with this image. And Sam and I have talked a lot about this and thinking about, well, how do you really get across to people how cool this might be? And so what you're looking at, I wish we could dim the lights. Maybe we can't. Well, what you're looking at here is a picture of, you can kind of see there's a bit of more of a bright stripe here and a darker stripe here. Oh, we can't dim the lights, can we? No. Can I here? Oh, it would be really, PhD. Oh. Ah, yeah. Is this okay with everyone? This will really be illustrative. So what you don't need to see me. What you're looking at here, oh, this is fabulous, is a picture taken by the Voyager spacecraft. It was 6.4 billion miles away, and I think it's 1990 if I get it right, Carl Sagan lobbied to have the camera of the Voyager spacecraft turned around. And he called this image the pale blue dot, because you are there. There. That's Earth. That's Earth from really, really, really far away as Voyager was leaving our nearest galactic neighborhood. And so Carl Sagan had a whole lot to say about this, but in particular, there's kind of a way to understand our place in the universe and this idea of what is life and why is life all the same, yet we think it's really diverse. And in particular, there's perhaps no better demonstration of the folly of human conceits than this distant image of our tiny world. To me, it underscores our responsibility to deal more kindly with one another and to preserve and cherish the pale blue dot, the only home we've ever known. And so when I think about what experiments I'm running, and we're a pretty complex series of experiments, humans themselves, ourselves, and a lot of cells, and all the cells are the same, but they're really different. You know, we can kind of play with those ideas. But the kinds of questions I think about, and that Sam and I have talked about, you know, and we can do these simple experiments and ask these simple questions. But what is it that really, one of the bigger questions that we all think of? So we think about where we are. Again, this is that Voyager spacecraft image. And I talked to my niece, she's 5, her name is Ashley. And you know, I may not be able to answer these questions, but she's gonna ask me 2 questions someday, and we don't know the answer to those questions. Are we alone? And how did we get here? And so if Sam and I can put our little peg on the board, and our collaborators in understanding how this microbe can do something maybe just a little bit different, what does that tell us about the life on our own planet and how it may have got here? And what insights might that give us for life on other planets? I don't know the answer to those questions, but it's profound to me that as scientists, we in the community, we don't know the answer to these questions. So we'd like to leave you today with thinking about how this tiny 1 by 2 micron microbe might be doing something really, really interesting, and yet in all other ways be something we might not think is very exciting. And to thinking about Earth 6.4 billion kilometers away, and that perspective of time and space, and kind of our role in the universe, and how we can think about our own planet and think about planets elsewhere. And with that, we'd like to thank you for your attention, and Sam and I would be happy to take any of your questions. Thank you. Oh, yeah. Did they get them? Yeah.
All right, so we're going to open the floor to questions, and I believe there's one right here, orange.
Yeah, the next logical place to look, if you start with phosphorus and go to arsenic, how about antimony?
Absolutely. We actually have a colleague at the US Geological Survey, because you know that microbe I described to you that can breathe in and out arsenic? There's a colleague of ours who's looking at antimony respiration, which is the same idea. So that's step 1, right? And then they're doing really neat new stuff on that. Yeah, thanks.
The next question will be over here in the center. So if you have a question, please raise your hand and we'll bring a microphone to you.
Have you isolated the nucleic acids from this particular bacterium? And have you seen the arsenic actually in that nucleic acid?
So we have done some preliminary isolation of the nucleic acids, and we show by mass spectrometry that we could actually see the arsenic there. Right now, what we're hot on the trail of is trying to do some more elaborate, more detailed studies of those kinds of isolations. But we're not just interested in the nucleic acids. As I showed you with that radio-labeled pie chart, protein arsenylation, well, that would be neat, you know. So we actually have some preliminary data, and we're trying to do that as fast as we can. Thanks.
Will the chemistry of the nucleic acids of this organism present problems in terms of the chemistry to sequence them?
It's an interesting question. You know, we have, I'm sorry?
Could you repeat the question?
Sure. I think if I got the question right, is the chemistry the same, similar enough between arsenic and phosphorus we could do the DNA sequencing?
Yeah, I'm just wondering if having the arsenic integrated in place of the phosphorus in the nucleic acids would present problems in terms of sequencing.
So the one thing that the evidence that Sam showed, and we don't have, of course, some of our over their tables, is that those bond distances are very, very similar. And that's one of the reasons that you saw that particular pattern. It turns out that it's similar enough that we wouldn't suspect it. We do have some colleagues that are going to do the genomic sequencing, so we'll find out, you know, how difficult that might be.
In the back left.
Thanks.
If arsenic is present in these microbes, is phosphorus at unusually low levels in these microbes? Do you have any microbes with practically no phosphorus in them after you've grown them for generations in arsenic?
Yeah, I think that's exactly true. So some of the measurements that we did, you really see that there's very little phosphorus. I mean, there's still some, you can scavenge, but it's definitely low. Definitely not.
But meaning too little phosphorus to be in between every base in the DNA sequence.
Correct.
If the bacterium that is extracted from monolake can incorporate arsenic, right? I mean, how come it's not naturally being incorporated in an environment that is heavily, you know, concentrated in arsenic?
So we don't know. We have no in situ, so we actually, one of the questions I could tell you that we have right now is how common is this in the environment? Right now, you know, we've isolated this in the laboratory, but we haven't yet had a chance to go back into high arsenic environments and see if this is actively being done by the microbes. Thanks.
Hi. So if arsenic can replace phosphorus, what else could replace phosphorus? And then my second question is, what are they doing differently, these little things, with arsenic in them as opposed to what they would otherwise do with phosphorus? In other words, has their behavior changed?
That's a pretty good question. So I would say, I mean, it appears from what we see in the chemistry at least that the arsenic is, you know, normally a cell would just want to get rid of it. It would store it somewhere else where it can't interfere or throw it out of the cell altogether. And this one's actually incorporating it. And it actually seems to be going in all the places that you would find phosphorus. That's our preliminary evidence. And so now we want to follow up with that to really see and break apart those areas and see, you know, each of these three fractions, the lipids, the proteins, the DNA, you know, what is arsenic doing in each of these cases that we see it there? And so like when you say other elements that could also do this kind of thing, so antimony was brought up as well. You want to look for things that are similar along that row, the column of the periodic table.
The only thing I'll add I completely agree with Sam. The only thing I'll add is that, you know, one of the things we like to think about, just like going back to the bioinorganic chemistry story we told at the beginning, is, you know, this isn't just about arsenic, and it's not just about Mono Lake. It's about asking questions in a different way of finding something that might have been in front of us the whole time. So arsenic is one example, and it has this relationship with phosphorus. But I think, you know, part of what we're interested in, and not just us, and what other people can bring to the scientific table, is what other questions can we start asking?
There's a question in the center of the back row. Okay, we'll start there.
Yeah. Why doesn't the arsenic work in the human DNA? Why does it work in the microbes?
You want us to take a stab at that?
I can take a stab. Why doesn't it work in the human DNA? So what I could tell you if I wanted to go into my normal, like, deep scientific lecture, which would be not really exciting right now, is that it turns out one of the things that really struck us a couple of years ago is that when scientists in the 1950s through about the 1970s were trying to understand why arsenic was toxic, and I told you that it was toxic because it looked like phosphorus, right? So when the scientists were working out that problem, what they noticed is that, let me think how to explain this. What they noticed is that some of the early steps of biochemistry, some of the early steps of the way like your body breaks down sugar, for example, seem to be able to incorporate arsenic. But then it's like, I have a new TDI Jetta wagon. It's really cool, but it's a diesel, right? So I'm really worried when I go to the gas station and I keep saying diesel, 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 because if I put in gasoline to my car, it'll work, but then bad stuff happens. Right? So I think of arsenic in our biochemistry as kind of the same thing. The first few steps of some enzymes will stick the arsenic on there, and then bad stuff happens. Right? And that's what I saw as a biochemist, you know, because I saw, for example, you're not going to know what this is, but Zen, you'll hear it again when you go to college. It makes glucose-6-arsenate. Oh my gosh. Hexokinase recognizes that and makes ADP-AS. Oh my gosh. So I really thought, well, that's really weird that certain chemicals our enzymes could make, and yet they couldn't complete the pathway. So it turns out, what you're really wondering, is that it turns out that those arsenic bonds, they're called esters, and the backbone of DNA, that's called an ester, like polyester, right? They're more volatile. They can break faster. So your biochemistry and my biochemistry is used to the bond having a certain lifetime, lasting a certain amount of time. So if arsenic gets incorporated, it breaks a little faster than our biochemistry is expecting. So even though, just like in his building blocks, arsenic could come in, that's what scientists showed, even before I was born, scientists showed this. Even though that was true, it's like the downstream processes. It's like my car putting gasoline in a diesel engine. It's the downstream processes that bad stuff will happen. And so, no, our biochemistry doesn't seem to recognize, but what I will put a bug in your ear about, because maybe you'll solve it someday, is that arsenic has been used for centuries to treat cancers. Now, it doesn't seem to me, when I read the scientific papers, that we really understand the mechanism of why arsenic has been used for centuries to treat cancers. Maybe the cancer is incorporated into their DNA and it has this lifetime problem. I don't know. But it's a good question, I think, to start asking. Does that answer your question? Excellent.
Okay. Two questions. One, with subsequent generations, do you see that these microbes prefer arsenic over phosphorus, you think?
Well, I think the question was, do they prefer arsenic over phosphorus?
After subsequent generations. If so, can it be used to clean up toxic environments?
Well, they can grow on phosphorus.
Yeah, and the growth curve shows that it prefers phosphorus still, even after 8, 10 dilutions, right?
Yeah, yeah.
So it would still like phosphorus if it was given phosphorus, but it can manage to do it without the phosphorus.
I wonder, I think there's a remediation possibility here for those who are a bit more applied. I think that's certainly a potential.
Okay. The other question is, today when we look for extraterrestrial life, we are looking for certain chemical signatures. Does it mean that we have to now look for different signatures also? Right. Because it seems like life can be without your regular chemical signatures.
That's an excellent question. Okay, I won't completely punt. I think that it's true. So we're looking for life elsewhere in the universe. And you might say, first, well, let's go back for a second. Why would you want to look for life elsewhere in the universe? Well, if we found life doing something different, what's the problem? The problem is solving the problem of being alive. That's the goal, be alive, okay? So if we found life different than us and yet solving that problem, you know, you could think of all sorts of things like biomedical implications, remediation, ways to take care of our own planet, that sort of thing. So I think one of the reasons for looking for life elsewhere is really practical. You know, it's another solution to the problem we cope with every day. So if we're looking for that life, looking for amino acids, you know, the building blocks of proteins, and we're looking for DNA, and you know, we know these things form spontaneously in the solar system. We know that there's amino acids in meteors, for example, and this sort of thing. So we'd like to think that our work, while it's preliminary and still there's clearly 30 years of us and all sorts of other people to bear down on this, it could give us this insight into life might be a little different out there if it could be a little different here. So, I mean, it's a really complicated problem, as you might imagine, measuring stuff on Mars, but stay tuned. We have colleagues at NASA, the Mars Science Laboratories, about to go up in November, and that'll get to Mars in the next couple of years, and that should be really interesting. They have all sorts of instruments on there that should be able to detect something a little bit different. But what does it tell us? To keep an open mind if we don't see what we think we know, right? Great question, thanks.
All right, 2 more questions.
So you mentioned that the bonds involving arsenic were weaker. Were there any properties other than the incorporation of the arsenic that you noticed that were different before and after the experiment, such as a shorter lifespan?
What was the lifespan?
They do grow slower. When they grow on arsenic, they grow slower. You saw it was kind of a slower uphill. The cells are you a biologist? Sort of, okay, that's good enough. So when you collect the cells, they're really soft compared to when grown on phosphorus. They're really robust. So they're just, yeah, they're really squishy, like really, really squishy. They're really difficult to work with. So we suspect there's probably really interesting morphology, so structural changes that we don't know. We didn't show it to you tonight, but we did some other kinds of electron microscopy. We took a thin section. It's like slicing you in a really thin section. We checked out all the pieces, and they look really different inside. And that's about as much as we know right now. Because we hadn't planned to do a whole study just like that. We just went, oh, that's really weird. Clearly, we need to figure out what's going on. But right now, we know that it's different. So what's kind of fun to think about is that when grown on arsenic, they look like I showed you, those pictures. And when grown on phosphorus, they look different, which means that if you look down on a microscope, it could be the same microbe, just in 2 different environments, right? So how something looks may not really give you insight into what it's doing.
Just real quickly, it was a cool story how you're diluting the stuff from one test tube to the next, and I'm sure a lot of cool stuff in science is not accompanied by a eureka moment, and so that's a very illustrative parable. Except, why were you diluting all this? That was the part I didn't understand. Like, what were you looking for when you found this? Was this what you were looking for?
Yeah.
Oh, okay, good. So that really was what you were looking for.
It was that question. Can a microbe grow? Can a microbe substitute arsenic for phosphorus the same as grow? Yep.
Just checking, because you sounded confused when you found it. And
well, no, the confusion was that while we have the hypothesis, generally you figure the null set will be proven. We assume it's not going to work. So when it does, it's really exciting, but you're confused.
All right, so I asked, and they're willing to answer more questions one-on-one, but so that some people can get out. It's been a while now. I'd like to end the open Q&A now, and before we go, let's thank our speakers one more time.
Please feel free to check out our microbial zoo.
Great job.